Here's a quick sketch of the area of the hippocampus where we're going to be looking. We have the entorhinal cortex, that's sort of the input, and you can think of the CA3 as some sort of associative memory structure, if you will. And you'll notice there's two paths from the EC to the CA3, and we're interested in this secondary path through the dentate gyrus, okay? This path is differentiated by the direct path because when you look at the signal going through it, there's an increase in sparsity and inputs are decorrelated. Okay, the common theory is that the dentate gyrus does some form of pattern separation. That is, two similar inputs come in and then they're sufficiently separated when they come out. Okay, and there's been an information theoretic argument that this action is necessary for proper formation of a CA3 network downstream. Okay, so that's what we're going to be looking at and modeling this coding behavior. Okay, we do want something that's rather neural inspired. So our general formation starts with a weight matrix, and we're going to compose that with a thresholding function. We have sort of a guiding light here in that we want something that's very simple combinatorially, but structurally rigorous enough that we can do mathematics with it, right? So we want to be able to formally prove all the claims that we make. That said, we want to see if we can, if we can have a system where we have all the properties that we want while having something that's very parsimonious, okay? And those properties that we want are the ones that we find in the literature about the dentate gyrus, meaning that it does that pattern separation, that the inputs become decorrelated as it gets processed. As a separation sort of from the sparse coding you see in other contexts, we want to make sure that the fidelity is something that we can control, right? We don't want it to be a byproduct of an iterative process or something like that. We want to be able to adjust it as we go, okay? And then once we have sort of a standard basis to start working with, what we're going to do is we're going to add biological constraints, and it's the neural inspired part of it will let the biology inform how we generalize our model to more sophisticated systems. Okay, so we do assume that our input space is n-dimensional. That's going to happen at the outset, and then we take delta to be a subset of the power set of the indices 1 through n. Okay, delta is going to be what's going to control our fidelity. So the larger delta is, the more fine-grained we have, and the smaller delta is, the coarser grained we are. And then we define our weight matrix where each of the rows corresponds with an eta I in delta. Okay, and the rows are going to be the columns are going to be positive if those indices are in eta I, and then we normalize over the size of eta I. And then we threshold at 1 because it's convenient. You can generalize that. But one thing to keep in mind for now is that the output dimension k is dependent on the size of delta, right? Because we have one row for each of the entries in delta. So just like as a quick example where we are, if delta is 1, 2, and 2, 3, and 2, 3, 4, then this first row in our weight matrix corresponds to the 1, 2, and things like that, right? The second row will correspond to the 2, 3. Once you see the example, I think it's fairly obvious. What happens is that since we're thresholding at 1, effectively each of the dimensions in the target space becomes like an AND gate over the indices in the corresponding eta I, okay? And for now, we're going to make one assumption. We'll be able to relax this later, but it helps us prove what we're working with in the meantime. We're going to assume that delta is composed of all p-size subsets for some p bigger than 1, okay? So with that formalism for now, we can show basically that it fits what we want out of the coding that the dentate has from the literature, right? So this first one here just says that outside of a neighborhood of 0, our mapping is injective, right? So that means there's some noise filtering around the origin, but outside of that, it's a lossless encoding. We can prove that we have a non-trivial decrease in the normalized dot products, that is the correlations. So this action does indeed non-trivially decrease our correlations. And then we can compute exactly the level of pattern separation that we have. Okay? Again, this pattern separation is important because if you think about it, if you're forming an attractor network or something like that downstream, you definitely want to have some separation between your storage points, right? The information you're storing. And like I said, we can guarantee a minimum amount. We can calculate exactly what it is depending on our parameters, NMP, and all that good stuff, okay? So that's sort of the basic situation. And from here, we're going to look to the biology and the neuroscience to see how we can generalize this to something that's a bit more realistic. I think this is really interesting because, you know, usually what you would do, you'd look to the computer science to make a more efficient coding, or you look to the mathematics to find a more convenient coding. But here we're looking to the neural side of things, and we're going to generalize it in a structured way that is informed by the biology. And I think that's a really interesting method. And what it's going to do is it's going to give us more realistic behavior. More is important. We're not claiming this is realistic to begin with. The assumption that I talked about with p-size subsets, we'll be able to relax that, and you know, we can sort of see how the model reacts to judge how useful it is. All right. We're going to talk about two types of generalizations really quickly. One, we're going to basically shape our fidelity over the input structure, and that's inspired by the grid cells in MEC. And the other one is adaptive fidelity via mixed coding method, which is inspired by adult neurogenesis. Okay. So the first one, it's sort of a classic idea. If you know the distribution of your inputs, well, you can shape the fidelity of your coding to match that distribution. Right? And in particular, if we remove stuff that we never see, that's just impossible because of the structure of our input, then you know we can do this this reduction without losing any information. Okay? So you know, talk this morning was a really good description of grid cells in the MEC. Just to refresh, they encode spatial information using this periodic modular code. Okay? It's a really interesting code because it carries an exponential increase in information capacity, and we want to respect that structure as it's going into the dentate. So what we do is we take our input space to be group isomorphic to this grid cell code, okay, or the grid cell input. And what that means, since it's group isomorphic, it's equivalent in terms of structure, but also in terms of operation. So those nice properties like the redundancy and the carry-free arithmetic and things like that are still reflected in this isomorphic space. And then we just remove the parts of delta that can't happen. And what results is that we can dramatically reduce the size of a target space, okay? And we can construct bounds for how large our target space will be because of this. It gets a little fuzzy because it depends heavily on the bounds, or sorry, on the periodicity and the spacing of your grid cells, but in general, we can still write good bounds for it. And what's really nice is that the literature has expectations for the activity sparsity and you know, the number of grid cells you have and how they're cast into the dentate and things like that. And when we apply this reduction to our model, it still fits those definitions, in particular with the expectations for sparsity. So that's really kind of neat. The other thing that we looked at is adult neurogenesis, which is new neurons appearing throughout life. This happens in the dentate, and the neuroscience literature says that young neurons are more broadly tuned than older neurons. And there's been some research showing that this helps increase information capacity, which sort of makes sense. Like if you look at the diagram, if you have a fixed coding, you can't really adapt to new information, or if old information becomes newly important, right? But by having a mixture of large and smaller receptive fields, you can adapt to the information as it happens, and we can incorporate this into the model. Namely, what we're going to do is we're going to take one of the entries in our delta, one of the A to I, and replace it with subdivisions of that subset, right? So we're further subdividing our previous entries. And they're technical, so I didn't include them, but you can, again, provide conditions that guarantee those results before, that still guarantee the increase in sparsity and the decorrelation that we want, okay? So just to recap, we have a nice simple combinatorial code, but because it's so simple, we can show that it has the properties that we expect out of the dentate from the literature. And then we've used the biology to sort of inform how we generalize and modify our code. And when we combine the two generalizations we talked about along with others, we actually have a really fine-grained control over the fidelity of our encoding. So that's about it. I do have a poster in that room. Should anybody want to look at it during the break, I'd be happy to talk about it. Thanks.